Everyone. Today we are discussing about the development of the nervous system. First, coming to the important points about the nervous system. The nervous system is derived from the ectoderm of the embryo and it is formed from the neural tube, and the peripheral nervous system is formed from the neural crest. That is, central nervous system is formed from the neural tube and peripheral system from the neural crest. The first cells to differentiate in the nervous system are the neurons, which are specialized for the communication. These are followed by the supporting cells known as the neuroglia or simply glial cells. Next, coming to the neural ectoderm. The neurons and the other cells of the nervous system develop from the dorsal ectoderm of the early embryo. Neuroectoderm, consisting of the neural plate, appears in the dorsal midline of the embryo at the 16th day, that is the third week after fertilization. The appearance of the notochord includes the overlying ectoderm to thicken and form the neural plate, and cells of this plate make up the neuroectoderm. Here is the picture showing the neural ectoderm, and here the cells will differentiate and will form the neural plate. Next, coming to the neurulation. Neurulation is the process whereby the neural plate forms the neural tube. The cells of the neural plate becomes taller than those of the ordinary ectoderm. This change is induced by the underlying mesodermal cells. The neural plate grows rapidly, and in the 2 days it becomes the neural groove with the neural floor along each side. And by the end of the third week, stage 10, the neural floats have begun to fuse one another, thereby converting the neural glue into the neural tube. This is the neural tube. This transformation begins in the middle. In that it will eventually be the cervical segments of the spinal cord, and the proceeds rostrally and then caudally. Then, coming to the neural force, until the fusion is complete, the cephalic and the caudal ends of the neural tube communicate with the amniotic cavity by the cranial and the caudal neurophore. This is the cranial and this is the caudal neurophore. The openings at the each end closes up at about 24th and the 27th days respectively. Then, coming to the neural tube. The neural tube is the forerunner of the brain and the spinal cord. The cells lining the tube constitute the neuroepithelium, which will give rise to all the neurons and most of the other cells in the CNS. This is the table showing the different time at development of the nervous system. That is, at the 18th day, neural plate will appear and thickening of the ectoderm between the primitive node and the oropharyngeal membrane occurs. And in the 20th day, neural groove and neural folds are formed. Neural groove is the continued thickening of the neural plate on its periphery, forms the midline neural groove with the thickened neural folds along the side of the groove. And at the 22nd day, there is the formation of the neural tube with cranial neurophore and the caudal neurophore. Neural tube, it is formed by the neural folds join in the middle to form the neural tube. Fusion of the folds proceeds cranially and caudally, eventually leaving a cranial and the caudal neurophore. And the neural tube separates from the surface ectoderm to lie between it and the notochord. Cranial part of the neural tube forms the brain, while the caudal forms form the spinal cord. And the lumen of the tube forms the central canal of the spinal cord and the ventricular system of the brain. And the cranial neurophore closes on the 25th day and caudal neurophore on the 27th day. Then, coming to the neural crest. The cells between the neural plate, this is the neural plate, and the ectodermal cells will give rise to the epidermis of the skin that are not incorporated into the neural tube. These cells come together in the midline to form the neural crest dorsal to the tube. Cells from this neural crest migrate laterally and eventually gives rise to the non-neural elements. The non-neural elements are as follows. The leptomeningitis, it forms the pia mater and the arachnoid, and cells of the autonomic ganglia, postganglionic sympathetic and the parasympathetic cell bodies, and cells of the spinal and cranial nerve ganglia forms the first order sensory cell bodies, and the Schwann cells form the myelin in the peripheral nervous system, and the chromaffin cells, neuroendocrine cells found in the adrenal medulla, and the melanocytes, pigment producing cells of the epidermis. And the pharyngeal arch skeleton forms the skeletal and connective tissue components of the pharyngeal arches. And then aorticopulmonary septum, connective tissue septum that divides the aorta and the pulmonary trunk in the heart. Then odontoblasts, cells that form the dentin in the teeth, and parafollicular cells, calcitonin producing cells of the thyroid gland. Then coming to the placodes. The techniques of the neural ectoderm give rise to the olfactory placodes and the otic placodes. The olfactory placodes, which form the cranial nerve 1 and induce the formation of the olfactory bulb, and the otic placodes, which form the cranial nerve 8 and the sensory apparatuses of the inner ear. Some of the neurons and the other cells in the peripheral sense organs and the ganglia are derived from the placodes, which are the thickened regions of the ectoderm of the embryonic head. Olfactory neurosensory cells, the sensory cells, and the associated ganglia of the inner ear, and some of the neurons in the sensory ganglia of the cranial nerves are derived from the placodes. Some cells of the olfactory placode migrate into the rostral end of the neural tube and become intrinsic neurons and the neuroglial cells of the CNS. Then coming to the neurons, the first populations of the cells produced in the neural tube are the neurons. Neuroblastic differentiation, migration, and the neuronal multiplication starts in the first 3 weeks of the embryonic life. Primitive cells destined to become neurons originate in or close to the neuroepithelium of the neural tube. These cells proliferate at an astonishingly rapid rate at 250,000 per minute according to the Common for the circumscribed period, that is several days to the weeks. They become transformed into the bipolar neuroblasts, which migrate in a series of the waves towards the marginal layer of what is to become the cortex of the cerebral hemisphere. The first glial cells also appear very early and provide the scaffolding along which the neuroblasts move. Each step in the differentiation and migration of the neuroblasts process in an orderly fashion, and one stage progresses to the next with the remarkable precision. The process of the neuronal migration is largely completed by the end of the 5th fetal month, but continues at a much slower rate up to the 40 weeks of the gestation. Most of the neurons are produced between the 4th and 20th weeks. The young neurons migrate, grow cytoplasmic processes, and form the synaptic connections with the other neurons. The number of the neurons formed in the neural tube exceeds the number in the adult brain and the spinal cord. Large number of the neurons die in the normal course of the development. This occurrence is known as the death of apoptosis. Cells that died were those that failed to make the synaptic connections. The neurons in the sensory ganglia are derived from the neural crest, send neurites into the peripheral nerves and into the neural tube. By the 8th intrauterine life, the centrally directed neurites have the extensive synaptic connections with the neurons in the spinal cord. The number and complexity of the synapses continue to increase until well after the birth, as does the generation of the neuroglial cells. Neuroglia, most commonly called glia, comprises the cells of the nervous system that are not neurons, and other than the neurons are called as glial cells. The first glial cells, known as the radial glia, develop along the sides of the first neurons, having the cytoplasmic processes that extend from the lumen to the outside surface of the neural tube. The process of the radial glia, it guides the migration of the young neurons. Most astrocytes and the oligodendrocytes, however, are generated from the neuroepithelium during the fetal period. Microglial cells arise from the hematopoietic tissue and enter the brain and spinal cord by passing through the walls of the blood vessels. In the peripheral nervous system, neurons, that is in sensory and autonomic ganglia, and the glial cells, that is satellite cells in the ganglia and Schwann cells in the nerves, are derived from the neural crest. Then let us coming to the formation of the brain and the spinal cord. After neurulation, the CNS is represented by the closed tubular structure with the narrow caudal portion and a much broader cephalic portion, characterized by the number of the dilatation of the brain vesicles. Then coming to the spinal cord formation, the spinal cord arises from the lower part of the neural tube, and the site of the closure of the caudal neuropore corresponds to the upper lumbar segments of the cord. Further caudally, the spinal cord is formed by the secondary neurulation, which is the coalescence of the chain of the vesicles that become continuous with the lumen of the neural tube about 3 weeks after the closure of the caudal neuropore. The vesicles are derived from the caudal eminence, a mass of the pluripotent cells located dorsal to the developing coccyx. Then coming to the neural tube wall and what they give rise to. The neural tube wall is divided into three layers: neuroepithelium, mantle cell layer, and marginal. Neuroepithelium, this is the ventricular layer, which is the innermost adjacent to the lumen of the tube. It is formed by the ependymal cells that line the central canal and the ventricles. Then coming to the mantle, that is the intermediate layer. This is the middle layer formed of the neurons and glia, gives rise to the gray matter. Then marginal layer, outermost, contains the nerve fibers from the neurons and the glia, give rise to the white matter. As the cellular proliferation and differentiation proceed in the neural tube, a longitudinal groove called the sulcus limitans appear along the inner side of each lateral wall. The sulcus demarcates the dorsal alar plate from the ventral basal plate. They acquire the afferent and the efferent connections respectively, and are present from the rostral end of the mesencephalon to the caudal end of the spinal cord. Responding to an inductive effect of the nearby notochord, which marks the position of the future vertebra, the basal plates of the left and the right side become separated by the thin floral plate. Next, coming to these are the alar plate, that is posterior, the lateral. It gives rise to the posterior horns, that is sensory, and the sulcus limitans separates the alar plate from the basal plate. Then the basal plate, that is the anterior and lateral, which gives rise to the anterior horns of the motor, and the roof, that is posterior covering of the central canal, and the floor anterior, that is the anterior wide commissure. Then coming to the additional concepts, the sulcus limitans is visible in the floor of the fourth ventricle in the adult brainstem and is a useful guide for separating the motor and the sensory nuclei. In an infant, the spinal cord extends the length of the vertebral canal. Growth of the vertebral canal outpaces that of the spinal cord, such that in the adult, the spinal cord only extends to the L1 L2 vertebral level. And some of the basal plate cells differentiate into the motor neurons with the axons that grow out into the developing muscles. The growing axons of the neurons of the sensory ganglia enter the alar plate. At the third month of the intrauterine life, the spinal cord fills the vertebral canal, and at the fifth month, the intrauterine life, the lower level of the spinal cord is at the L5 or S1. And at the birth, the lower level of the spinal cord at the level of the third lumbar vertebrae. Then coming to the development of the spinal nerve, motor axons growing out from the neurons in the basal plate. Sensory components arise centrally and peripherally from the growing fibers of the nerve cells in the dorsal root ganglion. Nerve fibers of the ventral motor and the dorsal sensory roots join to form the trunk of the spinal nerve. Then coming to the development of the brain, the three major divisions of the brain appear at the end of the fourth week. The three main divisions are the prosencephalon, mesencephalon, and the rhobencephalon. Prosencephalon is the forebrain, mesencephalon is the midbrain, and rhobencephalon is the hindbrain. And during the fifth week, the secondary swellings develop in the prosencephalon and mesencephalon. During the fifth week, the secondary swelling develop in the prosencephalon and rhobencephalon, so that the number of the major parts become 5. That is, prosencephalon divides into telencephalon and diencephalon, mesencephalon is the same, and rhobencephalon divides into metencephalon and myelencephalon. Next, simultaneously it forms the two flexures, that is the cervical flexure at the junction of the hindbrain and the spinal cord, and the cephalic flexure in the midbrain region. Flexures in the neural tube help to accommodate the initially cylindrical brain in what will eventually be a round head. These flexures in the brain ensure that the optical axis of the eyes are at right angles to the axis of the vertebral column. These are the different structures, that the primary vesicles and the secondary vesicles, which will give rise to which part of the brain in the adult. That is prosencephalon, that is the forebrain. It is divided into telencephalon and diencephalon. Telencephalon gives rise to cerebral hemispheres and the olfactory bulbs, and the ventricular system formed within it is known as the lateral ventricle, and the diencephalon gives rise to thalami, and the ventricular system is the third ventricle, and the midbrain, that is mesencephalon, gives rise to the midbrain. It is ventricular system is the cerebral aqueduct, and then comes the cephalic structure between these two, and after that is the robencephalon, which gives rise to metencephalon and the myelencephalon. Metencephalon gives rise to the pons and the cerebellum, and myelencephalon to the medulla. This ventricular system is the fourth ventricle. Then comes the cervical flexure and the developing spinal cord. This picture that depicts the different parts of the primary vesicles, secondary vesicles, and the adult brain structures. Then coming to the ventricular system, the lumen of the neural tube becomes the ventricular system. The lateral ventricle develops in the each cerebral hemisphere. The third ventricle is in the diencephalon, and the fourth ventricle is bounded by the medulla, pons, and the cerebellum. The third and the fourth ventricles are connected by the narrow channel, the cerebral aqueducts, through the midbrain. The lumen also remains narrow in the caudal part of the medulla and throughout the spinal cord, where it becomes the central canal. Then coming to the development of the meninges, the membranous coverings of the brain and the spinal cord first appear in the fourth week as a single mesodermally derived primary meninx. Full fluid-filled spaces appear within the primary meninx 1 week later, and the subsequent differentiation leads to the formation of the three layers that constitute the meninges: the pia mater, closest to the nervous tissue, arachnoid, and the dura mater. This is the video which shows the development of the nervous system. Let us see the video.
Neurulation is the process of neural tube formation and its development into the spinal cord and the brain. Any complication during this complex process can result in neural tube defects. The development of the nervous system begins during the third week of gestation. Primary neurulation is the process that forms the functional central nervous system. Let's begin with the development of the embryonic nervous system. By day 18 of fetal development, the ectodermal germ layer has the shape of a disc with a cranial and caudal end. The cross-section on the right illustrates three germ layers: the ectoderm, mesoderm, and endoderm. At this point, the central nodal cord has induced the overlying ectoderm to thicken and form the neural plate. The cross-section on the right will help visualize the folding of the neural plate, leading to the formation of the neural tube. By the end of the third week, the edges of the neural plate extend upward to become neural folds. The depressed mid-region is now the neural groove. Each neural fold then grows toward each other to fuse at the midline, converting the neural groove into a complete enclosed and hollow neural tube. At the same time, as the neural folds fuse, a population of cells separates from the tips of the neural folds to become a mass of mesenchymal cells called the neural crest. The neural crest cells migrate throughout the body to form a wide variety of cell types, including Schwann cells, the meninges, endocardial cushions, parafollicular cells, and the adrenal medulla. Before the fusion of the neural tube is complete, the cephalic and caudal ends communicate with the amniotic cavity through the cranial and caudal neural pores. The cranial neural pore closes on day 25. After closure, the cranial end of the neural tube develops into three primary vesicles: the forebrain, or prosencephalon, the midbrain, or mesencephalon, and the hindbrain, or rhombencephalon. These three develop later into 5 secondary vesicles. The forebrain develops into the telencephalon and diencephalon. The midbrain develops into the mesencephalon. The hindbrain develops into the metencephalon and the myelencephalon. The caudal neural pore then closes on day 27. During early embryonic stages of development, Failure of the neural tube to close properly results in neural tube defects, one of the most common causes of congenital abnormalities. Neural tube defects can occur as part of syndromes and associated with chromosomal disorders or as a result of an environmental exposure. Folic acid deficiency during pregnancy and folic acid antagonists such as carbamazepine.
Okay, next coming to the developmental anomalies of the nervous system. That is, the first one is the anencephaly. Failure of the cranial neuroploid to close may cause anencephaly, a serious birth defect in which the brainial and the cranial vault fail to develop. And then coming to the spina bifida, there are different types of the spina bifida. Failure of the caudal neuroploids to close may lead to the spina bifida, which includes the following variants. First one is the occulta. There is only the vertebral arch defect, and in cystica has two forms, that is meningocele and meningomyelocele. In meningocele, meninges project to the vertebral arch defect, forming the cerebrospinal fluid filled cyst. And in meningomyelocele, spinal cord tissue projects through the vertebral arch defect into the CSF field meninges. That is, in meningocele only meninges, and in meningomyelocele meninges along with the spinal cord. And in myelocystis, there is the open neural tube defect. These are the different types of the neural tube defects. First one is the rachiasis, and second one is the spina bifida occulta, and third one is meningocele, and this is the meningomyelocele. Only meninges, and here is the meninges with the spinal cord. Next, coming to the neurocrystopathies. They result from the abnormal specification, migration, differentiation or death of the neural crest cells throughout the embryonic development. Additionally, they arise because of the genetic defects affecting the formation of the neural crest and because of the action of the teratogens. This is the classification of the neurocrystopathies. That is, dysgenetic neurocrystopathies are pigmentation anomalies, abnormal angiogenesis, facial dysostosis, and 22q11.2 deletion syndrome, Charge syndrome, Hirschsprung disease. And neoplastic neurocrystopathies are neuroblastoma, pheochromocytoma, and paragangliomas, and MTC, and melanocytosis and melanoma, neurofibroma and schwannoma. MTC is medullary thyroid carcinoma. Mixed dysgenetic and neoplastic encephalopathies are familial neuroblastoma and pheochromocytoma, and paraganglioma and MTC. Combined congenital central hypoventilation syndrome, Hirschsprung disease, and neuroblastoma, and MEN2 syndromes and neurocutaneous syndromes. Next, coming to the congenital hydrocephalus. Hydrocephalus is the dilatation of the developing ventricles caused by the excessive CSF, typically resulting from the failure, that is the blockage of the ventricular drainage system to remove the CSF and move it into the circulation. The causes of the hydrocephalus may be inherited or acquired. The inherited causes are myelodysplasia, atresia of the foramen of the Monro, neural tube defects, that is encephalopathy, spina bifida, and genetic abnormalities like aqueductus stenosis, Dandy-Walker complex, and other cerebral and spinal malformation. And the acquired causes are tumor, meningitis, infection, hemorrhage, arachnoid cyst, posterior fossa cyst, and traumatic brain injury and idiopathic. And next, coming to the Arnold Chiari malformations. The Chiari type 1 refers to the displacement of the cerebral tonsils to the foramen magnum without dysraphism. And meningomyelocele combined with the inferior displacement of the cerebral tonsils through the foramen magnum is known as the Arnold Chiari malformation type 2. The other features of the type 2 malformation include elongation and thinning of the upper medulla, pons, and the bony defects of the foramen magnum, occiput, and the upper cervical vertebra. These are the, and type 3 and type 4 are the other types. In type 3, further herniation of the cerebellum below the foramen magnum, forming the encephalosphyle in addition to the spina bifida. And in type 4, hypoplasia and aplasia of the cerebellum with the spina bifida. Next, coming to the disorders of the prosencephaly, that is holoprosencephaly. The absence of the hemispheric suppression results in the single large forebrain ventricle is the holoprosencephaly. These are the degrees of the severity. And coming to the agenesis of the midline prosencephalon development leads to that is agenesis of the corpus callosum and septo-optic dysplasia. This is the agenesis of the corpus callosum, this picture, and this is the septo-optic dysplasia. Next, coming to the disorders of the neuronal proliferation. Decreased proliferation will lead to microcephaly or microlesencephaly, and decreased neuronal proliferation results in the microcephaly with or without the abnormal cerebral morphology. Birth head circumference is 3 or more standard deviations below the normal. Then disorders of the proliferation, that is hemimegaencephaly, and that is the unilateral enlargement of the cerebral hemisphere. Then abnormal neuronal differentiation or maturation. First is the tuberous sclerosis complex, and second one is the focal cortical dysplasia. And disorders of the neuronal migration, that is which will lead to the heterotopia, that is collection of the ectopic neurons located outside the cortex. Then lesencephaly, paucity of the normal gyri and sulci resulting in the smooth brain is the lesencephaly. And cobblestone malformation due to over migration of the neurons beyond the disrupted basal membrane and pial surface and onto the overlying subarachnoid tissue. That is, the brain tissue is lies below the subarachnoid tissue. This is the cobblestone malformation, gives rise to the cobblestone appearance. And disorders of the post-migranal development, impairment of the cortical organization after the neuronal migration, and polymicrogyria. Then schizencephaly, it is characterized by the abnormal slits or clefts in the cerebral hemispheres of the brain. This is about the development of the neural system and the defects. Thank you. This is about this presentation, and I will come up next with the different topics.